Hi everyone. My today's topic is structure and functions of biomembrane or plasma membrane. So in this we are going to see what is meant by plasma membrane and then what is the functions, how far it is composed with defects. So if there is any defect in plasma membrane, what are the disorders? These things we are going to see in my class. So specific learning objectives in my class. At the end of the lesson, the students must be able to describe the fluid mosaic model of biomembrane with a labeled diagram, functions of it, and what are the factors that increase or decrease the membrane fluidity, functions of various transport mechanisms across the biomembranes with suitable examples, and at last, what are the disorders associated with biomembranes. So why should we take care of plasma membranes? As biological membranes are thin, flexible surface separating cells and cell compartments from the environments. Problems with the cell membrane involved in many diseases such as type 2 diabetes. Type 2 diabetes, we can see what happens to the insulin resistance. If there is any defect in the pancreas cell membrane, insulin will not be produced. If insulin defect or insulin receptor defect, then it leads to hyperglycemia. That leads type 2 diabetes. An organ transplant rejection, very important thing in which when the donors and receptors, the particular membrane should be matched with the other donors or receptors. So if there is any defect in the plasma membrane, organ transplant rejection happens. And cholera, you'll be knowing, it is toxins may be introduced into the body if the plasma membrane is not properly working. Cystic fibrosis. So cystic fibrosis may happen due to thickened of mucous membrane, and cancer targeting plasma membrane lipid composition repair mechanism. It is potential strategy for cancer therapy. So these are the disorders if plasma membrane is defective. And these are the organelles containing membranes such as lysosomes, Golgi bodies, mitochondria, and nucleus. So next we'll move on to functions. Plasma membrane functions, selective barrier. It acts as a boundary controlling the substance which that enter and exit of the cell in order to maintain homeostasis. Transport, membranes protein like channels and pumps transport ions, nutrients and other molecules across the membrane. Cell communication is very important functions because cell-cell communication inside the cell, inside the subcellular organelle is very important that leads to metastasis of cancerous cells. So the membrane contains receptors that bind to external molecules, ligands initiating internal signals for cellular response. And cell recognition, carbohydrates plays an important role as cell surface receptors they are working, which is nothing but oligosaccharides. And last, structural integrity, it provides a flexible yet secure boundary for the cells and helps to maintain the cell shape. So these are the functions of plasma membrane. And just in a picturization I have given in order to easily memorize or recall. Transporter, enzyme activity, cell surface receptor, cell surface identity marker, cell adhesion, attachment to the cytoskeleton. So first we are introducing what is membrane. Membrane is for transport. It is cell membrane boundary between inside and outside. So what should be entered inside and what should exist out? Membrane will choose. That's why they are called as semi-permeable membrane or selectively permeable membrane. It can't be impenetrable boundary. In food, carbohydrates, sugars, proteins, amino acids, lipids, other gaseous as well as salts will be make an entry of it. Whereas out particles, ammonia, which is very dangerous and toxic substance that may enter into blood-brain barrier. So it should be existed out. Ammonia, salts, carbon dioxide, water, and the other products will be existed out. So now comes 5-mark question, fluid mosaic model. This is very important 5-mark question for university exam. So first you should write what is a definition of plasma membrane as I've told in the before slides. Next we have to tell about who proposed this model. Davison and Daniel in 1935 originally proposed a membrane structure. Singer and Nicholson described the structure of biomembrane as a fluid mosaic model. So here this is plasma membrane structure, 3D structure I have given, but in next slide I have given a simple structure that you can draw in a diagram for exam point of view. So in this phospholipid bilayers, hydrophilic head, hydrophobic tail. So these greens are hydrophobic tail, and this is hydrophilic head with integral proteins and extrinsic proteins. So extrinsic proteins attached with sugar particles, that is called glycoprotein. That's why it is commonly called peripheral protein. And integral proteins, I've given example inside with phospholipid bilayer. So this is overall structure of fluid mosaic model. So next, this is the structure you want to draw. You have to draw in the exam point of view. So I have shown nicely it is tightly packed. Phospholipid bilayers, in between cholesterol present here, which maintains the fluidity of the plasma membrane, and this is integral protein directly or closely associated inside the plasma membrane, in which it is acting as an ion channel protein to make the larger particles and charged particles to entry inside. Whereas other integral proteins also will be helping for transport and cell communication. And this is glycoprotein, which combine with the protein sugar particles, and this is protein. That's why they are called as glycoprotein. So overall, when you are drawing the diagram, you have to mention the parts also. That will give more marks. Presentation. So composition of the cell membrane, lipids. Lipids like phospholipids, sterols. Proteins like integral protein, peripheral protein. Carbohydrates like glycolipids and glycoproteins. So these are all the constituents present in the plasma membrane as a composition. Just a small recap, it is highly selective. Again, I'm telling functions are very important. It is highly selective and composed of lipids, proteins and carbohydrates. Phospholipid bilayer plays a major role, 90% of it composed in plasma membrane, creating a stable fluid bilayer. So next, lipids, small simple structure you have to draw side by side fluid mosaic model with hydrophilic head and hydrophobic tail. That's why they are called as amphipathic in nature, both water lovable and water hateable. So phospholipids, glycolipids and cholesterol in the composition of lipids. And proteins, already I have told, I'll be giving examples for integral proteins. Channels and receptors are embedded within or span the bilayer, while peripheral proteins are cytochrome C of mitochondria. So in MCQs, mostly they'll be asking examples of integral protein and extrinsic proteins. In that you can give the examples channels and receptors for integral and cytochrome C of mitochondria for peripheral. So they perform functions like transport, enzymatic activity and signal transductions. Carbohydrates 2 to 10% contributes by the sugar residues as glycoproteins and glycolipids, mainly for recognition and signaling process. Cholesterol, 2 mark important question. They'll ask how cholesterol maintains the fluidity of plasma membrane. So in animal cells, cholesterol acts as a fluidity buffer, stabilizing the membrane and preventing it from becoming too rigid or too fluid at different temperatures. So increased cholesterol, less fluidity on outer surface and more fluid in the hydrophobic core. Here I want to impress an important thing. Isomers, cis form and trans form, as we have read as a chemistry, cis form and trans form, what it is going to do in our body. So cis form increases the fluidity, prevents tight packing and makes a bilayer difficult to freeze, whereas trans form decreases the fluidity. For example, I want to give cisplatin and transplatin. Cisplatin in the form of same side isomer, it acts as an anti-cancerous activity for cancer patients. Whereas transplatin, it is not acting as an anti-cancerous because it is in trans form, whereas cis form is good. So as an isomer, what it is going to do, like that, don't think as a chemistry part, very important in biochemistry. Cisplatin and transplatin in anti-cancerous activity, very important examples for isomers. So recap of functions, it acts as a outer covering or just protect from environment of the cells, selectively permeable, homeostasis in order to maintain the balance, and it keeps a proper concentration of nutrients and water and eliminate the waste. And now comes functions, mechanisms of transport. Uniport, symport, antiport it is there. So in uniport, same direction or single compound, transport of glucose to RBC, just a function of plasma membrane. Symport, sodium and glucose transport in intestinal mucosal cells. Antiport, exchange of chloride and bicarbonate in RBCs. So in this case, you can tell sodium and potassium also as an antiport exchange. Simple diffusion, passage of water and gas Facilitated diffusion, glucose transport in muscles and adipose tissue. Primary active transport, sodium potassium pump. Secondary, co-transport of sodium and glucose. So these are all just functions I want to give. Later in other classes I have given our active and passive transport explanation, but in this class I just want to tell the structure, composition and functions. Next comes very important clinical applications of channels. Sodium channels, Liddle's disease, which is nothing but elevated osmotic pressure of blood resulting hypertension. So here in the case, sodium will be regained back, reabsorbed back. Sodium and water will be reabsorbed back, whereas potassium will be existed out. So that is called Liddle's disease, which leads to hypertension. Potassium channel, very important. Defect in heartbeat, convulsion in newborn, inherited deafness. So these are all important channels that works in our body. And chloride channel, cystic fibrosis, kidney stones may happen. Cystic fibrosis, very important 5-mark question. They may ask in the plasma membrane defect. So these are some of the disorders of membrane. Cystic fibrosis, red cell membrane defects, achondroplasia, congenital long QT syndrome, Wilson disease, eye cell disease, and optic neuropathy. Leber's hereditary optic neuropathy also may happen if the plasma membrane isn't defective. So cystic fibrosis, it occurs in European populations and they are the carriers. Obstruction of GIT and respiratory tract leading to bacterial infection of airway and death finally due to respiratory insufficiency before the age of 30. So these are the clinical features. As an MBBS student's clinician, you should write what are the clinical features and symptoms for each and everything. So respiratory signs and symptoms, digestive signs and symptoms, reproductive system I have given here as persistent cough, wheezing, breathlessness, exercise intolerance like that, and digestive, foul smelling, greasy stools. You have to give the important, much important signs and symptoms for each and everything. Reproductive system, vasa deferens, congenital bilateral absence of vasa deferens happens. That may lead to infertility. So these are the symptoms of cystic fibrosis. And this is very important point, mutation in the gene for cystic fibrosis transmembrane conductance regulator gene. If there is any defect in this mutation gene, then it leads to cystic fibrosis. Chloride channel will be disturbed, results in defective chloride transport. And this causes thickened mucus in the respiratory and digestive tract. Liddle syndrome, already I have explained. Long QT syndrome and Brugada syndrome, cardiac arrhythmias, disorders caused by mutations in ion channel like cardiac sodium channel or potassium channel that disrupt the electrical signaling in the heart. So this is human ether related gene. This is sodium channel with alpha subunit 5. So if there is any defect in this, it forms QT syndrome, lengthy QT syndrome. Whereas familial hypercholesterolemia, you will be knowing if there are defect in low density lipoprotein, that leads high bad cholesterol, which increases the risk of atherosclerosis and heart disease. And sphingolipidosis, membrane fluidity disturbance, these are all commonly we'll be learning in lipid storage disorders. Niemann Pick disease, neurodegenerative disorders it leads. Alzheimer disease, it is common for if there is any neurological symptoms happen, it is common now. Alzheimer disease happens, memory, reasoning, language, coordination, mood and behavior will be changed. So next, muscular dystrophies, condition like limb-girdle muscular dystrophy with the repair protein of dysferlin, which is essential for repairing the plasma membrane in muscle cells after injury. So lysosomal storage disorders, you'll be knowing clearly as in subcellular organelles we have learnt, and Chediak-Higazi syndrome, it leads to formation of abnormal large lysosomes and problems with immune functions and pigmentations. So thank you. So if there is any doubt regarding this, you can contact me in the YouTube channel by asking questions or any queries. Thank you, my dear students.